Welcome back to class, Curious Molecules. So today we are going to continue with carbohydrate metabolism. So if you have probably wondered once or twice, remember we are Curious Molecules on this page. So you have probably been wondering in your life once or twice, or as many times as you can't count, about how the body really works, how the body is able to break down large chunks of food. Maybe mechanically you know that your teeth do it, but how come you excrete all of that, but the body still has energy from somewhere to make you function properly? So today we are continuing with metabolism. We have talked about introduction to carbohydrates. We have talked about digestion, absorption, and we started with metabolism of carbohydrates. So if you haven't watched those videos, you should definitely go and watch it. Today we are continuing with another type of metabolic pathway that carbohydrates are involved in. Is how does your body survive when you have not eaten all day? If we know that food gives energy, how come there are people who could go 24 hours without eating? Or maybe you're so busy into your work that you forget to eat. You probably maybe even forget to drink water. So what is the backup system of the body? When food is not coming in, where does all the energy come from? So we are going to see today that the body has its backup energy source, like a backup generator, where it uses to process glucose. Remember from the previous discussions we have had on metabolism, we have seen that glucose is the primary source of energy production. The energy we get from ATP, which is a currency, is gotten from breakdown of glucose in glycolysis, which is the first and the most basic pathway that we saw last time. So now, what is this backup pathway? Gluconeogenesis. So we said the body has a backup source of glucose, and gluconeogenesis here can be broken down into three different words. The glucose, neo means new, and genesis, synthesis. So genesis from the beginning, from other sources, because glucose is also stored. We know that you can get glucose from storage, but if there is no storage at all, you have to create it from scratch, and that is what gluconeogenesis is. So the definition is basically forming or producing, synthesizing in basic terms, like in a factory, trying to produce glucose from gathering small materials or gathering other materials from scratch. When you're trying to produce a car from scratch, you gather some parts and then you put them together and they form that particular car. So with gluconeogenesis, you're gathering materials or intermediates from different pathways to get glucose from scratch. So in gluconeogenesis, those other pathways are like amino acids, fatty acids, and so on. You get those sources and then you produce glucose for your body to use. So in the body, only two organs have the ability to produce glucose from scratch or can perform gluconeogenesis. The liver, which does 90% of this gluconeogenesis process, and the kidneys, which does it as a backup. So we on Curious Molecules try to make all of these complex pathways as relatable as possible. So we are looking at gluconeogenesis in a kind of factory kind of light. We're trying to produce glucose, and the body is like the factory. So you're seeing the biochemical workshop here that is going to be responsible for making glucose. We are seeing the raw materials here, and we are seeing at the end, after gathering all these raw materials, they are exported and used for probably energy and functioning of the body. For storage, well, when the body is carrying out gluconeogenesis is because there are urgent needs by the body. So there will probably be little or nothing to be stored. But if there is, it's going to be stored. That's why we have a storage and a pantry here. And fueling the pathway itself, fueling the bakery. So the workshop itself, we will see that gluconeogenesis also uses energy. So we are producing energy to continue with, to fuel the body to continue with all the metabolic processes that are required by the body. So we already said that the raw materials for gluconeogenesis are gotten from other sources, which are amino acids, from fatty acids, and from lactate, which is a byproduct of anaerobic respiration. We're going to see for human beings. So we are doing that kind of analogy to better understand how gluconeogenesis functions. Is assembling glucose from scratch, and these are the raw materials in a certain factory in the body. The factory is the cytoplasm and the mitochondria of liver cells and kidney cells. So in the factory, you have a process, the assembling of the product. You get it from the raw materials and you begin to assemble them in a stepwise manner. And this is where you have the steps of gluconeogenesis, with specific enzymes that are required. At the end of the day, you have final assembly, which produces glucose. So this analogy is just for us to better understand this procedure. So from this, you see from our curious thinking of how the body is able to provide you energy even when you don't have energy coming in from food. It's a way to show that your body was meticulously created by your creator, such that every system has a backup system. And even the backup systems have backup. So we see that the liver is responsible for producing glucose. But if there is no liver, or if the liver is tired, if the liver is malfunctioning, if you are sick and the liver is affected, the kidney can do it for you. So there's a backup even for backups. So when your body has actually broken down completely, you know that it has really tried for you. So you should really tell that body that, though I'm sick, but thank you for doing this for me. And so you should also try to take care of your body. Try to feed it right. You should not eat too much of fat. You should not eat too much of sugars and all of that. You should try to do that. The body they try for you. All right. Okay, so as we have seen in the demonstration, the factory recycles other materials to produce glucose and is divided into sections or departments, which are the cells and the cell organelles that gluconeogenesis takes place in. All right, so now we are coming to the real core of the matter, the process of gluconeogenesis itself. In the factory, the assembly steps. So at the beginning, we have non carbohydrate molecules like amino acids, like fatty acids. And these are going to bring in molecules that go into the process of gluconeogenesis. So, in diagrammatically, we see that gluconeogenesis is a reverse of glycolysis. But we'll go further to explain when we go ahead that it's not simply like, okay, at the end of the day, we've had glycolysis, then we reverse it back, take the glucose back to, take the results back to glucose if you have to produce glucose. It's just writing it out, it looks like that, but maybe practically, glycolysis does not just finish and then gluconeogenesis takes over to reverse it. No, that's what they mean by it is simply not a reversal. So it occurs in reverse. The steps are in reverse, but it is simply not just a reversal. Because of that, and also because there are reaction steps that are bypassed in gluconeogenesis that we see here in pink and we see in blue for glycolysis. So for glycolysis, the end product is pyruvate. But pyruvate here is not directly converted back to phosphoenol pyruvate as in glycolysis. We see that it goes through two different steps. It's converted to oxaloacetate, and this particular step requires energy. The enzyme is pyruvate carboxylase. And from oxaloacetate to phosphoenol pyruvate, you have another enzyme, pep carboxykinase, which also requires energy. And from there, you have the rest of the pathways follow the same pattern as glycolysis. They are basically synthesized by the same enzymes. So you would also have the production of NAD. So you also have these two last steps that are bypassed. Bypassing means they are synthesized by other enzymes that are not the same for glycolysis. So at this step, from fructose 1,6 bisphosphate back to fructose, you have fructose bisphosphatase 1. So this fructose bisphosphatase 1 is an ATP required step. ATP is also involved, it's broken down here. And then from glucose 6 phosphate back to glucose, you have glucose 6 phosphatase. So these are the reactions that are different for gluconeogenesis as compared to glycolysis. As we explained before, it is simply not a reversal. As I said, glycolysis does not just end and then gluconeogenesis takes over. It's only the steps to explain it theoretically that we see that, okay, the steps follow it as if they are just a reversal of glycolysis. And also the steps also are more than that of glycolysis. The enzymes required for 4 steps, the bypass reactions, are different for glycolysis. That's why we say they are not simply just, the step is simply not just a reversal of glycolysis. And by the way, we saw here that oxaloacetate is involved here. But where does this oxaloacetate come from? Oxaloacetate is found in the mitochondria. This oxaloacetate is found in the mitochondria, but this part of gluconeogenesis occurs in the cytoplasm. the conversion from oxaloacetate to glucose. It occurs in the cytoplasm. So there is a procedure that takes oxaloacetate from the mitochondria to the cytosol so that it can continue with the reaction. Normally, oxaloacetate sits inside the mitochondria like a queen that cannot be exposed to our hash sun, so it has to be carried from there to the through the mitochondria into the cytosol. So it is carried by a shuttle called the malate shuttle. So this is inside the mitochondria. Oxaloacetate is sitting pretty and it cannot move on its own into the cytosol, so it needs a shuttle to carry it through. So it is moved by malate, so it's converted to malate basically, and malate can now move out of the mitochondria into the cytoplasm. When it's moved to the cytoplasm, it is reconverted back to oxaloacetate. Then the oxaloacetate can continue the rest of the steps. So this is one important aspect of gluconeogenesis. The reason why it occurs in two different compartments of the cell is because there is one of the assembly particles that are needed, but they are not present where the assembly goes on. So it is supposed to be moved from that part of the cell to another compartment of the cell, and it cannot move on its own. So there is a shuttle that carries it through the mitochondria to the cytoplasm. Oxaloacetate is converted to phosphoenol pyruvate, as we see here. Oxaloacetate to phosphoenol pyruvate, and it is an energy dependent step. But here we are seeing GTP and GDP. So in the comments, I want you to tell me what GTP and GTP are and how they relate to ATP. All right. So we already said phosphoenol pyruvate to fructose 1,6-bisphosphate. The normal glycolytic pathway with all the enzymes that synthesize it before. The fructose 1,6-bisphosphate phosphatase enzyme, which catalyzes the fructose 1,6-bisphosphate to fructose 6-phosphate. It's like a regulatory gate. It's a gate which helps to decide whether or not glucose production should continue. Remember when we talked of glycolysis, we talked of regulatory enzymes. Enzymes that if the body doesn't want glycolysis to go on, it's going to reduce the functioning of those enzymes, the activity. So here, fructose 1,6-bisphosphate is that kind of regulatory step. Let's say after that fasting you started eating, the body was still producing glucose through gluconeogenesis. So now you've eaten food and the body has extra source of glucose. So the body has stopped gluconeogenesis from happening. It's going to reduce that fructose 1,6-bisphosphate activity by downregulating its expression at the gene level. The body does not want glucose anymore because there's extra source from external sources, so it's going to reduce it. All right. So the final product is our free glucose, which is released by glucose 6-phosphatase. We already said that only the liver and the kidney are capable of producing glucose from scratch. If a particular part of the body is able to produce a particular molecule, it means that it has what is required for that molecule to be produced. If it's able to produce, if the liver is able to produce glucose, the kidneys are able to produce glucose, meaning they have a particular molecule that other cells do not have that can produce glucose. So it's like having a key to the gate that can unlock that molecule. And that's what the liver and the kidneys provide. So we have seen a summary of gluconeogenesis. Remember, as we said in glycolysis, we're highlighting key pathways here because we're not going deep into biochemistry. We're talking about biochemistry in a relatable way. And here we're looking at the energy cost of this pathway. In the body, when you're even in glycolysis, when we were doing glycolysis, we saw investment phase and the energy production phase. So in the body, every pathway has that investment. You're trying to produce glucose that will give energy for certain parts of the body, but you still need energy to produce the glucose. So there is an energy cost of producing glucose, and it's quite more expensive than even glycolysis. So we have the 4-2-2 rule. In the production of glucose, we have 4 ATP molecules that are involved here. The ATP molecules are used in the pyruvate carboxylase step, 2 ATP molecules. 1 ATP molecule is used here in the fructose 1,6-bisphosphatase step. 1 ATP molecule is used in the glucose 6-phosphatase step. So that is altogether 4 ATP molecules. Then we have GTP too. Remember, I asked you to check in to write the relationship between GTP and ATP. ATP is used in this carboxykinase step. And then you have NAD, which is also produced, sorry, which is used at this step here. So that's the 422 rule in gluconeogenesis, the cost of energy that is used here. We are going to see later in another metabolic pathway how NADH is vital in energy production. We are here to continue to trigger our curiosity towards our body. So while learning to understand the pathways, we can also question some of these pathways, how things go, what if they went the other way, and so on. So for you, in addition to what you have understood or not understood in this pathway, you should comment if you could redesign this pathway. We have seen that this pathway has complex usage of energy. What would you change in this particular pathway? That's how science works, basically. You ask questions and then try to answer them. All right, so regulation, which is what we don't fail to talk about in every metabolic pathway. Here we have allosteric regulation. We have not defined what allosteric regulation is, maybe, or maybe we have. I think we should have in one of the previous videos. Regulation of metabolic pathways by the products of that pathway or molecules involved in that pathway. Biology would discover that in the active site of an enzyme, or in an enzyme, there's an active site and then there's an allosteric site. The allosteric site is where other molecules bind to regulate the enzyme. So pyruvate carboxylase, which is an enzyme involved in gluconeogenesis, would have an active site where the substrate will bind, but it also have an allosteric site where other molecules can bind to regulate it. Pyruvate carboxylase would be regulated by ADP, by AMP, citrate. Phosphoenol pyruvate carboxykinase will be regulated by coenzyme A and acetyl CoA. So we are going to, in the comment section, give explanation of what these molecules mean, how this, try to explain how this regulation occurs, because we have explained it in glycolysis. So to test yourself, try to explain to yourself what this means. And if you can't, of course, then for you in the comment section, write your comment or your question. So there's also regulation by compartmentalization of this gluconeogenesis pathway. So we said that it occurs in two compartments or two parts of the cell, the mitochondria and then the cytoplasm. So the first step where we have to move oxaloacetate from its pretty chair in the mitochondria to the cytoplasm, it's a type of regulation because if there is no movement, then nothing will happen. So the take-home message is that gluconeogenesis is the process of synthesizing or production of glucose from scratch. From scratch, we mentioned before that you're getting glucose from sources which are not carbohydrate sources. You're using sources like amino acids, fatty acids, to produce a carbohydrate molecule, which is glucose. We have not seen any involvement directly of amino acid in the pathway. So how do they come into this pathway? We see here from these highlighted molecules or these circled molecules that some of them are amino acids. ASP, for example, is aspartate, phenylalanine, and so on. So some of these amino acids are incorporated or converted directly to molecules that are involved in the cycle of conversion to oxaloacetate. And oxaloacetate is an immediate precursor of gluconeogenesis. So that's how amino acids contribute in this case. And we see amino acids like glycine, alanine, which are directly converted to pyruvate. Pyruvate is also a direct precursor of the gluconeogenetic pathway. So we see how amino acids are incorporated into this. In addition to amino acids, we can therefore say that these other molecules that we see on this table are substrates for gluconeogenesis. If they are available, then during starvation, then gluconeogenesis will go on in the liver or the kidneys. So now we see pyruvate and lactate. So lactate is a byproduct of anaerobic respiration or breakdown of glucose in the absence of oxygen would produce pyruvate, which is converted to lactate. So when there is excessive lactate, the lactate can be reconverted to pyruvate to go into the gluconeogenetic cycle. Also, we see propionate, which is this substitute here, or this substrate here, alanine, which is also from breakdown of muscle, and glycerol, which are fatty acids. So for glycerol, we'll go back a bit. It's not indicated here, but glycerol is a substrate that is directly converted to this dihydroxyacetone phosphate. Glycerol is converted to this, and of course, it's converted to glyceraldehyde 3-phosphate, which is part of the intermediates in the gluconeogenetic cycle. So this is how all of these substrates contribute to the synthesis of glucose from non-carbohydrate sources. And as we said on this page, we are trying to be curious, trying to learn the body, understand the biochemical aspects of what is happening in our body, break it down into simple, understandable ways, and also try to relate it to what we see that is applied in medicine, applied in therapeutics, and all of that. So here we are taking note of an aspect of gluconeogenesis, which is a key contributor to hyperglycemia in type 2 diabetics. So in the comment section, I want you to attempt an explanation to this. Why is it a key contributor to those who have type 2 diabetes? Thank you for watching and thank you for liking the video. I hope that you understood at least a certain percentage of what we were trying to explain today. I hope gluconeogenesis seems easier to you. And if you have any questions with regards to this topic or other metabolic topics, don't forget to write in the comment section below. Also, comment the answers to all the questions that have been asked in the video. Share this video with people whom you think might be of help to, and subscribe to get more on this channel.